Good morning. We know that cortical areas are characterized by a specific site architecture and receptor expression pattern, and that they also differ in their functions. And in particular, primary sensory areas are characterized by very specific functional processing mechanisms which differ from those of hierarchically higher areas. And our working hypothesis was that primary sensory areas would also show a very specific receptor expression pattern, and that this expression pattern would be an evolutionary conserved aspect of the brain. And we applied the method of quantitative in vitro receptor autoradiography to test this hypothesis. So we examined up to 16 different transmitter receptors. And we exposed our radioactively labeled sections with microscales, which are those little squares that you can see up above the section. These standards have known concentrations of radioactivity, and they enable the computation of a regression curve, which together with the parameters of oh God, right. Sorry. So together with the parameters for the binding protocols, which are the specific activity and the association constant of the ligand used, would enable the transformation of gray values in each one of your pixels of your autoradiograph into a concentration of radioactivity. That's the image with the black background at the bottom of the slide. And these concentrations can be then transformed into color codes just for visualization purposes. So this slide here shows a coronal section through a human occipital pole, and the section has been labeled to visualize the muscarinic M2 receptors. Red codes for high receptor densities and blue tones for low densities. And you can immediately see that the primary visual cortex stands out by its much, much higher M2 receptor density than the adjacent secondary visual cortex, V2, or any other of the cortical areas at this level of the brain. And if we examine this same level of the brain, but with other receptors, you can see that differences in receptor densities, either over all the cortical layers or just in specific layers, enable the definition of the border between V1 and V2. And most specifically, the alpha-2 serotonin receptors, the 5-HT2 receptors, show specific differences, as well as the GABA-A and the nicotinic cholinergic receptors. If we move from the primary visual cortex into the other two primary sensory systems, this coronal section again through the human brain at a more central level, showing the central sulcus at the top of the section and the Heschel gyrus in the lateral fissure, you can see that again the M2 receptors show these two primary sensory areas very clearly due to their extremely high densities. For the primary auditory cortex, we have again receptors which show both borders of this brain region, such as, for example, the 5-HT1A or the nicotinic receptors, but the metabotropic glutamate receptors do not show either of the borders of the primary auditory cortex. And for the somatosensory cortex, we have the same situation. So some receptors, such as the alpha-2 or the nicotinic, show the borders of primary somatosensory area 3B, but other receptors, such as the GABA-B, don't. Now, summarizing the results of receptor distribution patterns in human primary somatosensory, primary visual, and primary auditory cortex, we can say that only 4 receptors, which are the ones highlighted in green, the muscarinic M2 receptors, the nicotinic alpha-4 beta-2 receptor type, the serotonin 5-HT2 receptors, and the adrenergic alpha-2 receptors, show the borders of all three sensory areas. And other receptor types show the borders either at specific laminars, layers, or in a couple of cases we have no borders. Now the next question that we asked ourselves in this study was if this characteristic pattern is evolutionary conserved. And in order to do this, we examined, apart from the human brains, the brains of 8 other species from different families, different animals that are commonly used in as models in neuroanatomical research. And the back of this slide shows the brains of some of the species studied, just to show you how different they are in size. And these differences in size I have not taken into account in the next slides, because I've blown up the brains of the small species to be able to show the receptor distribution patterns more clearly. So back to the primary visual system, back to the M2 receptors. The left of the slide shows the primary visual cortex in humans, and right next to it the macaque monkey brain, and you can see that the situation in the macaque monkey brain is the same as in the human. So the primary visual cortex stands out by its extremely high M2 receptor density. If we move to the rat brain, we can see that the M2 receptors also clearly show the border between V1 and the adjacent cortex, but this time not due to overall differences in receptor densities, but only to changes which happen at the level of layer 4, which is highlighted by the asterisk in this slide. And in the marsupial brain, we see a completely different situation, which is that V1 can be identified by differences in M2 receptor densities, but in this brain it doesn't stand out due to its higher densities than the surrounding cortex, but exactly by the opposite situation, by lower densities. And concerning the rodent brain, you might think, well, okay, these differences you find between rodents and primates aren't surprising, because the rodent isn't characterized by being a visual animal. Rather, it uses its whiskers to examine its environment. But if we move into the primary somatosensory cortex, we find exactly the same situation, which is basically that in the rat brain, differences between par 1, the par 1, the barrel cortex, and the surrounding cortex aren't found overall in this area, but also, again, only in layer 4, which is again highlighted by the asterisk in this slide. And again, in the marsupial brain, we see that the primary somatosensory cortex is identifiable not by its higher densities than the surrounding cortex, but by lower densities. So again, summarizing results and concentrating on the 4 receptor types, which in the human brain were all significantly different in the primary somatosensory cortex, primary visual cortex, primary auditory cortex than in the other areas, we can see that the only receptor type which across evolution remains a standard as a marker for all 3 sensory areas is the muscarinic M2 receptor. Now, the other aspect that we also examined was the differences in laminar distribution patterns, and this is also an important aspect to take into account when choosing your animal model for your scientific question is differences in laminar distribution patterns. And what we see here is that the serotonin receptor is an extreme case which in primate brains is characterized by very high densities in layers 1 and 2 and much lower densities in the deeper cortical layers. This already changes when we move to the scandensia, to the tree shrew, the tupaya brain. Here we find that actually the deeper cortical layers show higher densities when you're in primary sensory areas than the superficial layers. And again, the rodent brain is a completely different situation, because here the serotonin 5-HT1 receptor is homogeneously distributed throughout the cortex. So in conclusion, we can say that the high muscarinic M2 receptor densities in primary sensory areas, in the primary visual, auditory, and somatosensory cortex of mammalian brains is an evolutionary conserved aspect which enables us to identify these areas due to its considerably higher densities in the primary sensory areas of eutherian brain and its lower densities in the marsupial brains. And that we have to take these considerable differences in laminar distribution patterns into consideration when we're using animal models to examine the function of primary sensory areas in primate brains and those of other species. And with this, I'd like to thank you all for your attention, and if you have any further questions or want to see more images of the other species examined, you're welcome to do so at my poster, and the standby time will be this afternoon. Thank you for your attention.
Why only M2 gets conserved, not the others, like those species?
Well, possibly because the M2 receptor for acetylcholine, that modulatory neurotransmission, is more important to regulate brain activity than the more or the faster reaction that you find via excitatory or inhibitory neurotransmission.
Hi, thanks, it was fantastic. Obviously, the receptors have certain functions in terms of their excitatory or inhibitory potential, and it seems like a good potential for having something like an excitotome or inhibitome of the brain if you look at the sort of relationship between receptor density and other measures. Mm-hmm. Is that something in terms of mapping that you're looking into, or?
It turned out that the M2 receptor is the evolutionary conserved one, but we never just look at one receptor time. We routinely examine up to 20 different binding site densities, and do consider these balances between the different excitatory, inhibitory, and the modulatory ones. So yes, excitotome, inhibitotome is a nice terminology to use and
everything's
to introduce.
Right now, one follow-up question, since it's also expressed in the heart, the M2 receptor, have you looked at the heart in all these animals as a sort of control to see whether it's completely conserved?
No.
In the sort of cardiac?
Interesting question, but no, we haven't.
Sympathetic system?
No.
Quick question. Have you considered doing this type of analysis in sensory areas that are a little bit more pesky, like vestibular, primary vestibular, or, God forbid, olfactory?
It is something that we can look into. I haven't started doing them yet. We are looking at other brain regions, though, and also comparing the primary with the hierarchically higher areas.
Doctoral students in the lab looked for the vestibular cortex in humans and in macaque monkeys, and also the vestibular cortex has a relatively high M2 receptor density, well delineating the vestibular cortex at the edge of the insular cortex and the operculum. So this is the primary vestibular cortex, and there is a patch of very high M2 receptor density.
In the macaque monkey brain that we've seen. In the rodent we haven't.
In the rodent not. this is much more difficult regarding the localization of the vestibular cortex in the rodent brain, the precise localization, but in the human brain it's also.
Yeah. So